In this video, we're going to learn about one of the most important concepts in biology and chemistry and how it relates to the concentration of substrate and situations where we have more substrate per unit of volume outside of a cell compared to the inside of a cell. Let's forget about the concept of a cell membrane for a second and just look at a simple dissolving reaction. So what we've done here is we've added a single polar molecule that dissolves in water. Glucose is an excellent example, and we want to see what happens to this glucose as it dissolves into this beaker of water. Now, for a simple question to begin, do we expect that all of this glucose is going to remain in the top left corner of this beaker? If you said no, you have a good grasp of common sense. Now, we can use our knowledge of polarity to understand that water is going to use its hydrogen bonds in order to pull apart the glucose molecules, and because water is moving faster than solid glucose will, the water would cause the glucose to separate, but that's actually not the whole picture, and we can explain how glucose will go from this to this just by looking at the glucose itself. For example, if we look at this glucose molecule up here, if it tries to move back here, it's going to hit this glucose molecule, and that's going to knock it forward like this. If this glucose molecule tries moving down below, it's going to hit this glucose molecule and be pushed away in the opposite direction like this. And likewise, if it tries moving down here, it's going to hit the glucose molecule below it, be pushed to the top, and eventually it's going to make its way into this area of the water, or this volume of the water, where there are no glucose molecules before. This also applies to all of the other glucose molecules because, again, if they move backward, they're going to hit the glucose molecules behind them, bounce off of them, and be pushed into this area of the liquid here. This is relatively common sense, but again, it takes a little bit of careful analysis in order to explain why we would expect the glucose molecules to spread out and adopt relatively equal spacing throughout the liquid like this. Now let's go back to the concept of concentration. So we can define substrate concentration as the amount of a given substrate, in this case glucose, divided by the volume of liquid that amount is spread out through. So if we look at this area of the beaker, this volume of the beaker rather, we can say that this is an area of high glucose concentration because we have a lot of glucose in this specific volume, whereas this area where there is no glucose, we can define as an area of low concentration because there is no glucose in this volume of liquid here. And what we can see overall is that over time, the solute particles that are dissolved will move from the initial area of high concentration and spread out into the volume that has a comparatively lower concentration. And this movement from high to low concentrations is what we call diffusion. And the sooner you begin to understand how diffusion happens, the more phenomena in biology will be able to make sense. For example, if we look at diffusion in the context of the cell membrane, we can see here in the outside space outside of the cell, we begin with a very high concentration of solute, and we have conversely a very low concentration, so low that this concentration would be 0 because there is no solute inside of the cell. So logically, for the same reason as the glucose diffusion in the beginning example, we would expect that as long as this solute is able to pass through a phospholipid bilayer, we would expect the diffusion of these particles to move this way from the outside of the cell to the inside of the cell. This phenomenon where we have clearly defined areas of high concentration and low concentration is what we call a concentration gradient. And again, this is a difference because we can clearly see that we have more solute on the outside than we do inside. This difference exists between substrate concentrations between two different locations, one outside the cell and one inside the cell. Although again, as we saw with the beaker, we can have concentration gradients that form in a single solution like this. Now, as we have observed here, as long as there is a noticeable concentration gradient that exists, diffusion will naturally occur over time, even so much as having a solute diffuse through a semi-permeable membrane, such as the cell membrane that we see here. Now, if we wanted to think of examples of solutes that could do this, the most obvious examples are oxygen and carbon dioxide, because they're small and nonpolar, which means they can pass through the space between the phosphate heads and go right through the nonpolar tails, because all of these are nonpolar. However, when we consider polar substances, diffusion is still able to happen, but requires a little bit of assistance in order for these substances to make it through. For example, if we think of water as a particle that is able to diffuse, we know that water cannot pass through the nonpolar part of the cell membrane, so therefore it requires some sort of assistance in order for this to happen, such as, in this case, a channel protein, which has the polarity that matches the substrate. So in this case, because our substrate is water, we would expect the inside of this channel protein to be polar, and therefore allow water to make hydrogen bonds with the inside and pass through this channel through the process of diffusion. However, with larger substrates, we would expect that you would need a much larger transport protein in order to get them through. So examples of large polar substrates, one possible example is glucose, because it's very obviously polar, and most amino acids as well. Even with non-polar R groups, amino acids still have the amine and acid group that make them polar, and because they're probably too large to fit through a channel protein, we would expect carrier proteins to be able to carry them through. And again, carrier proteins, of course, require a specific active site, because, for example, a carrier protein for glucose would not be able to carry amino acids through it, because the active site has a specific structure. So here, we can see diffusion is still taking place, because in the case of a small polar substance such as water, although ions are actually the most common ones, we can see that these are still flowing from an area of high concentration outside the cell to an area of low concentration inside the cell. So this is still diffusion, but it is diffusion with a little bit of assistance, which is why we call this facilitated diffusion. To facilitate is a verb that simply means to assist or to provide guidance in order to be able to do something. And the structure in the cell membrane that does the facilitation of the diffusion are, of course, different examples of transport proteins that we see in the case of both channel or carrier proteins used below. In the next video, we're going to look at a situation where we have movement of given substrates, but where the movement moves in the opposite direction as our concentration gradient, meaning if we want to move substances from a low concentration to a high concentration, what would we need to be able to do in order to get particles to move in a seemingly unnatural direction of flow?